during this day. It's a symposium which deals with novelties in biomaterials and regenerative medicine. And although this may, we will see, at the moment, not ready in routine clinical therapy, it is our belief that it's extremely good for a clinical audience to get insight into the current research trends. In short, we have invited one of the top world scientists in the field of regenerative medicine. That is Professor Sam Stoop. Sam will give a short overview of the recent data, the current update on nanotechnology and regenerative medicine. After that, we have two Swedish stars, two young scientists working in cell biology and material science. The first one is Håkan Engqvist. He is developing new ceramics. You all know about ceramics in the clinic, but with some added features. Thirdly, we have Anders Lindahl, Professor of Clinical Chemistry and Regenerative Medicine here in Gothenburg, who is actually a pioneer together with his clinical collaborators in the field of autologous cartilage repair. So with these three excellent scientists, we hope that these 1.5 hours will be thrilling for you. So after some initial disturbances of technical character, I would like to welcome Sam to the podium, and please enjoy, everyone. We have questions after each lecture.
Good morning. Thank you, Peter, for the opportunity to be here this morning. My lecture is going to be about the design of bioactive nanostructures for regeneration. And this audience here probably doesn't need to be reminded, but I will anyway, that the humans are living longer and wish to do so for 100 years with high quality of life. And this is why regenerative medicine is an extremely important field in this century. So it will be just a matter of both sustainability as well, because we'll need to keep large populations in more functional, both physically and mental state in order to survive better. The field of regenerative medicine is, of course, highly interdisciplinary, so in that sense, it is a great opportunity for science. And nanoscience, I have believed for some time, is a key element that could play an important role. So the typical targets when we think about regenerative medicine are those that you see on the slide. And you can, for example, imagine that there are many problems here that are not solved. For example, how to deal with spinal cord injury. The regeneration of the central nervous system is an enormous challenge, as is heart regeneration. And of course, curing neural diseases like Parkinson's and Alzheimer's disease. These are all part of the regenerative medicine portfolio, let's say. Very relevant to the meeting here, of course, is finding the correct strategies to generate new cartilage and to have the strategies to deal with all bone fractures in a universal way. So the idea that we have developed over the course of the last 10 years or so was to think about a nanostructure that would be highly one-dimensional in the form of a fiber, of a cylindrical fiber that would mimic collagen fibrils, and that would self-assemble in situ around cells, display the signals, and then have the capacity to promote processes that are important in regeneration. This schematic here shows the concept in a highly magnified way. So here's the nanostructure. It is a cylindrical, one-dimensional nanostructure. It is, because of its internal structure, and is made up of many molecules. So you see here the detail implies that this is a supramolecular nanostructure. It forms by self-assembly of small molecules. And so the self-assembly is molecularly designed so that the fiber, the nanofiber, can display signals perpendicular to the long axis of the fiber that can directly talk molecularly to receptors on the cell surface. So here's the cell surface, there are the receptors. The nanostructure has the capacity to communicate with the cell to promote signaling. And so the molecules that we developed about 10 years ago for the first time to do this are called peptide amphiphiles. And so we were the first ones to develop these molecules designed to form the high aspect ratio cylindrical nanofibers. And so the notion here is that you have molecules with a peptide sequence that is covalently grafted to a hydrophobic tail. So the self-assembly of these molecules in situ, right around cells, would take place due to formation of hydrogen bonds among the peptide units, and then the hydrophobic collapse of these alkyl tails, which don't like to be at the interface with water. So this assembly that should occur in situ would display any signals that are at one terminus of the molecule here would be exactly displayed on the surface in very high density, and I should say, in fact, in tunable density. Now, the trigger for this process is the fact that there are always charged sites in this molecule. So when these molecules in water are injected in a physiological environment, the electrolytes in the medium naturally screen those electrostatic charges, and then self-assembly is triggered. So the typical structure in real chemistry is shown here. The red portion would be the signal for regeneration. And then there is a very important domain here in the middle, which is what we developed as the strategy so that when these molecules, as shown by this computer simulation, begin to interact in a electrostatically screening environment, they form, this particular domain leads to the formation of these beta sheet structures, which are very stiff. And of course, the beta sheets are tethered at the ends, as you see here in gray, by these hydrophobic alkyl tails. And now the collapse of those beta sheets that form upon self-assembly leads to the formation of the cylinder. And so the interesting thing about the platform is that we can basically put any signal that's desired here and then still trigger, because of the beta domain, the formation of this filament or very high aspect ratio nanostructure. Here you see by cryo-electron microscopy what they look like. They have extremely high aspect ratio, and they are very uniform in diameter, as you can see here, and also extremely long relative to their diameter. So the diameter might be 6 to 10 nanometers, but the length could be on the order of microns, and even in some cases that they could achieve macroscopic lengths. So these nanofibers, which bundle, for example, in some cases, as you see here, these bundles are in fact sometimes ordered arrays. We recently published a paper in Science reporting on the crystallization of these nanofibers. And so they look basically like a network that you might expect in the extracellular space. So they are mimics, basically, of collagen-type fibrils or fibronectin fibrils. And, but of course, they can be customized with nearly infinite diversity to display any signals that are needed right on the surface. And of course, the fibers logically can hug cells very easily, can network around cells, and stabilize themselves that way. So the platform is extremely extensive. So here you just see 10 examples of different molecules. They all form the fibers. This slide also illustrates the notion that you can take two molecules, the short one and a long one, for example, and then co-assemble two of them, not just one. And so now this expands the platform very much. In this case, the long molecule happens to have a peptide sequence, which we discovered in our lab to bind specifically to BMP2, a very important growth factor in bone regeneration. So the platform is extremely extensive. You know, we have probably made about 400 molecules over the past 10 years, demonstrating that you can customize these molecules and create a very, very large array of materials for regenerative medicine. I'll start by illustrating the application of the platform in neural problems. So here we focused on this signal, IKVAV, which is a signal that's present in laminin protein and binds to integrins, and is very important. When it binds to integrins, this signal causes neurite sprouting. So it's a way to elongate axons to create branching in neurons. And as we showed in this paper, also this nanofiber with high density of IKVAV signals, which by the way are on the order of 10 to the 15 signals per square centimeter when every molecule has a signal. They can also, as shown in this paper, lead to the differentiation of neural stem cells into neurons, which is very important in many of these CNS targets listed there. So these molecules bearing this sort of neurite sprouting signal are being used in our lab in a model of Parkinson's disease, for example. So we do a stereotactic injection directly in the brain, in the substantia nigra, where the dopaminergic neurons are. So a liquid comes in, and then during electrostatic screening in the brain environment, the filaments self-assemble, displaying very high densities of this laminin signal, which is active during our brain development. And so you can see, for example, pretty remarkable effects behaviorally in the animal model. So here you see the Parkinsonian mouse. So this is a mouse that has many of the symptoms that you see in humans during the development of Parkinson's disease. We induce this behavior by injecting the drug reserpine, which depletes the brain from dopamine. And so you see typically these three Parkinsonian mice. In the next shot, you will see how the Parkinsonian mouse compares to a perfectly healthy mouse shown here. So this Parkinsonian mouse dies always at about day 8 or day 9. When the nanofibers are injected in the brain, then the mouse does not die at day 8. This one, for example, has been alive 150 days, and this is as a result of a single injection of the IKVAV presenting filaments. A single injection in the brain. The mouse also recovers from the Parkinsonian symptoms, and as shown in this particular slide, a very interesting observation is that when we inject in the brain the peptide, as opposed to the peptide amphiphile filament with its capacity to self-assemble, then there's a huge difference in behavior. So in the case of the IKVAV, pretty much as you see from this axis right here, the mice basically die like the control at about day 8 or day 9. When injected with the self-assembly molecules to make the filaments is when they lose the Parkinsonian symptoms over time and also survive and live pretty much a normal lifespan. So we're very excited about developing this particular structures, this family of structures, perhaps as a neuroprotective therapy to alleviate the symptoms or the progression of Parkinson's disease. You see here when we harvest the brain in the substantia nigra where the filaments have been injected and induced to self-assemble, a lot of Cajal stain with gold particles shows that there's a lot of neural activity in the injected side relative to the side that is not injected. So another example in the field of bone regeneration. So here the design is a little bit different. Rather than having a laminin signal, as you just saw in the first example, we make use of residues of a phosphorylated serine, because this amino acid residue occurs, I mean, the basis of the design is that this amino acid residue occurs in many of the proteins that are responsible for mineralization in bone and also in dentin. So with these filaments, we discovered that the nanofibers, which you see here now, pretty dark by electron microscopy, and that's because when exposed to osteogenic media, for example, including alkaline phosphatase and glycerol phosphate as a source of phosphate, they nucleate hydroxyapatite crystals, but the interesting thing is that they nucleate them with the C-axis orientation parallel to the long axis of the filaments. And this basically is biomimetic of the orientation of the C-axis of hydroxyapatite in bone relative to the collagen fiber axis. So this is a biomimetic observation with these filaments containing the phosphorylated serine residues. And so in an in vivo model, a rat femoral critical defect model, we have recently seen, and this paper was published just a few weeks ago, that in the defect region, this is the control right here, when we simply place this nanofiber network in the gap, we see extensive bone regeneration after a period of about 4 weeks or so. And this is just placing those filaments with the phosphorylated serine residues, which we know nucleate hydroxyapatite. There are no growth factors added here. There are no cells added. This is just bone regeneration that we have shown very clearly. It's linked to the presence of the nanoscale filaments. This is the quantifying the amount of bone in the defect. And in this particular case, we have combined two bioactive residues. One is the phosphorylated serine for nucleation of hydroxyapatite. The other one is this fibronectin RGDS signal, which is responsible for cell survival and many other processes. And so in this case, we see the most bone formation comparable to the positive control, which is demineralized bone matrix. So with just a small molecule, we are able to pretty much induce the bone regeneration over a relatively short period of time without using any growth factor at all. Another example on cartilage repair. Here we have used in the animal model this microfracture technique, which is well known clinically, and where the bone is drilled and is caused to bleed. But now the environment of this bleeding bone is going to trigger the self-assembly of the filaments of the nanofibers, and then create a network, effectively a gel-like structure, forms very rapidly over the defect, and then we examine cartilage regeneration in time. So this is the paper that reports on the results. It is a filament with two molecules. It combines two molecules. So one molecule has this active, bioactive epitope, which we designed and discovered by phage display techniques to bind strongly to TGF-beta1, an important growth factor in cartilage regeneration. And the rest is just sort of a filler molecule. They both co-assemble, and they display this tether, which binds to TGF-beta1. So here are the results, and which are quite interesting in the defect that this is 12 weeks, and we've used a rabbit model to test this effect. You see here at gross examination of the tissue after 12 weeks, you see nice regeneration of cartilage, this sort of glistening white tissue. And histologically, in the defect site, so this is a cross-section, here is the defect area, you see very nice regeneration of cartilage with chondrocytes in columnar structures, lots of proteoglycan staining, And the system is this one here, in which 1 out of 10 molecules in the filament has the capacity to bind TGF-beta 1. In this case, you see very not great repair. This is mostly collagen. So the blue is collagen. And what we've done here is, in fact, used a filament that doesn't have any bioactivity per se, but we have added TGF-beta 1. So we've used the growth factor. Here, we have not added the growth factor. So this regeneration here is typical of a system in which the only thing that has been used are 2 small molecules, no growth factor, no cells, and the O'Driscoll score, which is in the paper, if you're interested, is up in the 20s. So it's pretty high quality tissue that has been formed. Okay. Another example that I can give you is the one where we have designed the filament to promote angiogenesis. So angiogenesis is just a process that is generally interesting in regenerative medicine. For example, in the treatment of myocardial infarction, it would be a very interesting approach to use a pro-angiogenic material. And so the way we've done this is by using heparin, the biopolymer heparin. We designed the peptide amphiphile to bind specifically to heparin, display loops of heparin, so that when small amounts of growth factors are added, for example, FGF2, then FGF2 can bind to heparin loops through the heparin binding domain, and this orients the protein better towards the signaling domain that binds to the receptor, and then this causes the angiogenic signal to assemble. These materials, which sort of basically empower the growth factors like FGF2, VEGF, lead to extensive growth of blood vessels within about 7 to 10 days in the rat cornea. And if you were to use, in this case, we've made this happen with FGF2 and VEGF, but with minute micromolar quantities of the growth factors, or even nanomolar quantities. When you use those tiny amounts of the growth factors, you see absolutely no growth of blood vessels. But when you use that same amount of growth factor with the heparin displaying nanofibers, then you see extensive growth of the blood vessels. This is generally an important target for many regenerative problems. This is one example. This paper is actually in press, but is not shown here. And so this is the use of this proangiogenic system in an infarct mouse model. And we see that after a month, we get the ventricular contractility functioning pretty much the same as a healthy control. And when we treat animals with just growth factors after the infarct is induced, we see no difference relative to no treatment. So again, here, it is the proangiogenic activity that we believe is causing the functional recovery. Very exciting new direction that we have is where we have actually, rather than designing to bind growth factor, we have actually designed to be a growth factor mimic. And so that's a very different approach. And we've selected the use of VEGF because the VEGF is a very important growth factor, as many of you know. It's involved in proliferation, migration, survival, angiogenesis also. And so it's an extremely important target. And VEGF requires a dimerization of the ligand and the receptor in order to be effective. So what we've done here is basically created a system where there is a molecule in the co-assembly that actually mimics the growth factor, and this peptide is from the literature, was discovered probably maybe 5, 6, 7 years ago, and it was reported to have the possibility to mimic the growth factor. So what we've done in an in vitro assay is to demonstrate that this system, which we call the VEGF peptide amphiphile, actually leads to the phosphorylation of the kinase that is supposed to be part of the VEGF signaling cascade. And most excitingly, that phosphorylation level seems to be higher than that induced by the growth factor itself and by and also the peptide, which is just the peptide alone without the without incorporation into the filament. So the system has been used recently in a model of hind limb ischemia. So as you know, this particular target is important because in diabetic patients, when there is peripheral vascular disease, then oftentimes this leads to amputation. And so it's very important to find ways of restoring flow, restoring blood flow. And as you see here, it's exciting to just follow when we inject in the muscle, just injecting in muscle the molecules that make the filaments, the VEGF-PA, just the peptide by itself without the nanostructure, and then some mutant form of the filament, which should not be bioactive, and then just the saline control. And you see here that the VEGF-PA filaments are the ones that gets us the highest perfusion ratio, measuring perfusion in the ischemic versus the non-ischemic limb. And so we have also evidence that, in fact, there has been a lot of angiogenesis promoted in this case, and we believe this is the source or the reason, the explanation for the restoration of perfusion in the system. Again, no cells, no growth factors. It's simply a filament that mimics the growth factor, but what's interesting is that in many ways it seems to be more effective than just the injection of the growth factor alone. There are systems that we have studied, as this one right here, with the fibronectin epitope, which show a very interesting effect that they lead to and this is now in vitro, they promote extensive proliferation of cells. In this case, these are mononuclear bone marrow-derived cells, and they can be when the cells are surrounded by our filaments, they actually, we observe enormous proliferation of these cells, and also when we co-inject those cells in tissue, then of course they're trapped by the filaments, but they also lead to, the filaments then induce proliferation as well. The importance of this slide, which you see how strong the effect is relative to when you just use a non-bioactive filament or when you scramble the sequence so that it doesn't have the proper fibronectin sequence, then you don't see the effect. So that tells you that you need the filament and you need the signal. And you need to be at a certain, we've shown before that in this particular signal, that level that molarity of signal in the filament is optimal for proliferation. And the importance of this is to suggest that this is a technology that might be a very good companion technology to all cell therapies. There's a great interest for many targets to use cells and using cells, but of course the problem is that you inject the cells and the cells swim away. Here the filaments would trap the cells. This paper actually shows that. And then of course, at the same time, it leads to proliferation of the cells for more effective efficacy. I'm going to finish up to just give you one example, since time seems to be running out. I'll finish up with this, which is just a sort of a teaser, as we say in the US, and it's just to show you what might be ahead in this type of platform. So in 2008, we published this paper demonstrating that these molecules, these peptide amphiphiles, can co-assemble with biopolymers. We use hyaluronic acid as the example in the paper, but this works with many other biopolymers. When they are oppositely charged, like negative and positive, and the charge density is carefully, very carefully adjusted, then you can observe the following process of self-assembly. This is a self-assembly that occurs on 40 millisecond or less scale. Time scale is very, very fast. And so in the lab, you take a pipette and deliver on a piece of glass. So this is something that you can see. This is real time. And this is the peptide amphiphile in water. Here's another water solution of hyaluronic acid, fluorescently labeled. And when the two water droplets touch, then instantly, in millisecond time scale, you form a membrane, a sac, which has completely engulfed the droplet of hyaluronic acid solution. And this membrane that you see here is a very complex membrane. If you're interested in the structure, you can read the paper in 2008. It forms a very complex membrane structure of fibrils that are parallel and fibrils that are vertical. And of course, what's interesting is that it is an excellent way to encapsulate things inside this membrane. A very interesting opportunity is shown by this curve right here. This is, by the way, the SEM of the membrane. You see the vertical fibrils that it has. And then on the outer surface right here, so this is the sac. And on the outer surface, you have peptide amphiphile filaments, the ones that we've been talking about since the beginning of the lecture. So that means I can cover the sac with any bioactivity, any specific bioactivity that I might be interested in. But the other thing that's interesting is that, as illustrated by this curve, this membrane is able to be permeable, is permeable to proteins. In this case, this is TGF-beta 1. So anything that's encapsulated in the center, in that green liquid that you saw, then comes out through the membrane. So this will become an interesting vehicle to deliver proteins. And also, the interesting phenomenon that we want to explore is to miniaturize this event of self-assembly so that we can make artificial cells. So basically make these sacs, not macroscopic as you saw them, but make them miniature in micron scale so they could be like artificial cells. Now this could be a very interesting vehicle to carry all kinds of things. On the surface it might carry real cells because it could have a bioactive carpet on the outside. On the inside it could have proteins or genes that can be delivered across the membrane. And here you see our recent efforts on microfluidics where we make the artificial cells, and this basically is the exact same process you saw before, except that this is now on the micron scale, and we're rapidly making all of these artificial cells that take the form of a microcapsule, and of course we can design them to be highly bioactive. I'm going to show you one example, and I think this is my last slide. Two. First, I just want to show you what the microcapsules could look like. This is one of them. This is a real skin cancer cell. And you see here this Velcro I was talking about. These are all the nanoscale filaments on the surface which can be designed for specific bioactivities. Here are all these other cells that we have made, and some of them emulate the architectures of red blood cells. When you, on the surface, if you make them bioactive, for example, remember the IKVAV signal I started my lecture with. So we've made these microcapsules with IKVAV nanofibers on the surface, and then we bring them in contact with real neurons. And it turns out, so here's the capsule, here's the artificial cell, and DAPI staining, you see the nuclei of the neurons that are sitting on the surface. They love the surface because it displays this laminin epitope, and then they start growing neurites, and actually we know already that they can make synaptic contacts. And so the neurons are being basically colonized on the surface of the artificial cell. And so this could be a very interesting way to deliver cells, for example, or to deliver from the inside the growth factors to the surface because there's permeability in order to differentiate for example, neural stem cells that might sit on the surface of the capsule. So this is just an example of a more complex platform where the nanotechnology builds into a micro technology, but that is much more complex, but still designed at the nanoscale. Okay, so I finished my lecture by taking the opportunity to thank my students and postdocs, since they're the ones that do all of the work, and the agencies that support our work in the lab. And finally, thank you, Peter, for the opportunity to speak, and many thanks to all of you for listening. Thanks.